Hi class! In today's video, we will be covering cellular respiration and metabolism. Cellular respiration occurs inside the cell, and more specifically in the cytoplasm and mitochondria. Essentially, the goal of cellular respiration is to take macromolecules from food, in this case glucose, and convert the chemical energy stored in the bonds of glucose into a form of energy that the body can use. The body's fundamental usable unit of energy is known as ATP, or adenosine triphosphate, and there's one major method that the body goes through to achieve it. That would be cellular respiration. Now, all organisms go through some form of cellular respiration. In eukaryotes, the main way would be through the mitochondria. But prokaryotes can actually use their outer cell membranes to do the same processes even without the mitochondria. So there are two types of respiration that our body utilizes, and that is completely dependent on one thing: whether or not oxygen is present. Firstly, you have aerobic respiration, which involves the later parts of cellular respiration, being the electron transport chain and the Krebs cycle. This process produces much more ATP but takes some time and needs oxygen. In the lack of oxygen, the body can use anaerobic respiration to generate quick ATP, but lactic acid buildup in the muscles can be really painful.
So this is our equation for cellular respiration, and it is quite simply the opposite of the photosynthesis equation. So we'll take glucose in the process of oxygen, and we're going to catalyze it down into carbon dioxide, water, and generate some ATP out of it. Now let's look at a little more into where the energy sits in this glucose molecule, because it's quite small and a simple molecule. How do we get energy? The energy is converted via the bonding and unbonding of hydrogens, and through the process, hydrogens fall off and oxidize with the oxygen we breathe in to make water. This happens because oxygen is hungry for electrons, so it takes them in from the hydrogen, and that affinity is where we get the energy to synthesize ATP. So this affinity creates a lot of energy, and the mitochondria control the reaction to make them more efficient.
So, the first step in the conversion of glucose would be glycolysis. Now, glycolysis is found in every organism on Earth and is the fundamental way to get ATP. So the essential goal is to split glucose into two 3-carbon pyruvate. This process takes 2 ATP to do and generates 4 ATP for a net gain of 2 ATP. Also, the splitting of glucose creates high-energy electrons and hydrogen ions that will be combined with NAD+ to make NADH. Now, NADH is a high-energy electron carrier and will be later used in cellular respiration.
Now that pyruvate molecule diffuses into the mitochondrial matrix and combines with the pyruvate dehydrogenase complex to turn the 3-carbon pyruvate into 2-carbon acetyl-CoA. Now that one carbon that was taken out is given out as carbon dioxide. So there is 1 carbon dioxide produced here for each pyruvate, 2 for each glucose.
Now, this acetyl-CoA goes into the Krebs cycle to be further oxidized. The whole point of this is to continue removing carbons and produce high-energy electrons that will be harnessed. Now, this acetyl-CoA cycles through the Krebs cycle, and each acetyl-CoA will create 2 carbon dioxides, 2 ATP, 6 NADH, and 2 FADH. Now, each of these high-energy electron carriers is going to move on to the final step of the process that will generate ATP. This is called the electron transport chain.
Now the electron transport chain is the most important part of the process here, and all the energy that was in the hydrogens of glucose is now either ADP or stored in NADH or FADH2. So looking closer, the electrons from NADH are released into the cytochrome complex and move through each of the proteins in the complex. The moving of the electrons causes the protein to pump one H+ ion that was released in the matrix by NADH into the inner membrane space. Every time an electron is passed through each cytochrome protein pumps a proton out. And what ends up happening is that there are a ton of protons that are filling the inner membrane space. That is the region of the mitochondria between the inner membrane and the outer membrane. That creates a chemical gradient with a high concentration of protons in the space and few in the matrix. But there is no place where those protons can go other than through a special enzyme, which is the final part of the ETC. ATP synthase, and this is a site of ATP synthesis. As the hydrogen ions flow back down the gradient through ATP synthase, the enzyme uses the energy in that flow to bind together ADP and a phosphate. This is known as oxidative phosphorylation, and ATP synthase acts as a rotor that spins together ADP and phosphate as the hydrogens flow through. and this process creates anywhere from 32 to 34 ATP, and the hydrogens and electrons that went through the ETC combine with O2 that you breathe in and become water. So oxygen is the final electron acceptor.
If there is no oxygen present, aerobic respiration is not possible. There is no oxygen to pull electrons. Glycolysis is good in that you produce 2 ATP by creating pyruvate, but that also creates NADH from NAD+. What eventually happens is that there is not enough NAD+ that is being recycled back. This causes a halt in the glycolysis process and the cell will die. Cells can go through lactic acid fermentation, where the pyruvate turn into lactic acid and then take the electron from NADH, thereby recycling the system. Lactic acid buildup can be painful and needs to be removed because it is toxic. That process requires oxygen. It can be a major problem if there is no oxygen to remove lactic acid or an enzyme is needed to break it down. Alternatively, alcoholic fermentation works similarly, but the pyruvate is broken down into ethyl alcohol and that releases carbon dioxide. And this is the reaction that yeast goes through when fermenting.
So, that's a basic overview of cellular respiration. Thanks for watching the video. Hope it helps. Bye class!